Oh, hey guys. All right. My name is Jack. I'm one of the TAs. So I'm gonna do a little workshop here, a little online workshop. If y'all like this, let me know. I really liked come up with like the information that I'll be displaying in this like a lot. But it was time consuming also. So if this is something you guys would like in the future, let me know. I'd be happy to. I just would, yeah, just let me, talk to me on the group me and whatnot, whatever. Yeah, so I found like a couple questions from like from MIT, like about like an MIT open coursework class for molecular biology that I think is relevant to some of the stuff you guys are doing in 514 right now. Yeah, I just want to talk about it. It's fun to talk about. And yeah, so this is like also I guess like part of like this is basically like biochemistry club sponsored workshop. All right, so that's kind of what these are going to be about. So whatever, let's get to work. All right, so I have like a couple questions set up. So this is the first one. All right, and we have it right here. One second, wait. Oh, okay, oops. All right. So here we go. I guess I'll read it. So you have isolated a spontaneous mutation in the leucine 6 gene of a haploid strain of Saccharomyces cerevisiae. Love that model organism. Fantastic, really. Some of your mutations result in a complete loss of leucine 6 gene function. You reason that this phenotype may be due to an insertion of a mobile DNA element in the gene. Very cool. Interesting. To test this idea, you sequence the gene from several mutants and compare the DNA previously determined sequence for leucine 6 in the parental strain. Numerous strains do in fact carry insertions, and by quick analysis indicates that several different types of mobile DNA elements are represented in the group of mutants. You wish to determine if the insertion mutations are caused by a cut and paste transposon, a retrovirus, or a DNA virus that inserts itself by conservative site-specific recombination. 2A. Below is the sequence of the mutation. The sequence of the parental DNA and the insertion is known based on this data, whatever, what is it? All right, cool. So here, let's look at this for a sec. So we know what a cut and paste transposon is. All right, we've heard of that before. A retrovirus, maybe not. All right, that retrovirus like HIV or something like that starts out as RNA, then uses reverse transcriptases to like make cDNA. And then from there, it starts making like lytic stuff and killing the cells. But we don't know much about that. Dr. Myers hasn't talked about that. But we do know about the DNA virus that inserts by a conservative site-specific recombination. That's like lambda, or like like a bacteriophage or just, well, it wouldn't be a bacteriophage in this case, right? Or lambda, because we're talking about Saccharomyces cerevisiae, which is not bacterial, right? So it's going to probably be some sort of like integrative virus that works like along the same lines. So you know what I mean? So yeah, so we need to be able to figure out if it's that or basically the cut and paste transposon, because me giving you the question is probably not a retrovirus. All right, whatever. So here are the two pieces of DNA, right? So this is the wild type, and then this is the wild type. Well, no, not the wild. It's the mutant phenotype. Right. And so it's the mutant phenotype. And so basically you have the normal gene and the like portions in the lowercase, and then the capital letters are representing the big gene portions. Like, or don't let me say the insertion. All right. So I recommend pause the video. I know you probably reading this and you probably don't know what the answer is. Probably intimidated, but I guarantee you, with the information have learned from Dr. Myers' presentations, once once you figure it out, you're gonna be like, oh wait, I could have done that. Like, makes sense. You're trying to figure out if it's a transposon or if it's site-specific recombination DNA virus. So think of what are we looking for if we have this situation. So I'll give you a second. Pause the video, I think. All right, are you on pause? Cool, welcome back. Whatever, maybe you didn't pause it, who cares? So let's get to it. So there is a pretty distinctive way to determine if it's a transposon or site-specific recombination of them. And some of y'all probably understand what it is, right? So if I think something is, if I see a mobile piece of DNA like this, like, and I think it's a, like, if I'm trying to figure out if it's a transposon, I'm looking for something in particular. I will look at the inserted DNA segment right here, and I'm going to look to the left and right of that. Here and here. And what do I see? I see duplicated sequences. So that is a very big indication of transposition. Because remember, and I'll show you the mechanism in a sec, but remember that if you see transposition, there's going to be two identical, like, you know, duplicated ends, all right? I want to see exactly where that is, like, where, I want to see where the if it, you know, assuming it's a transposition event now, a transposon, I want to see like where here it went to, right? So I'm looking for GCCT. GCCT is here, but also see it right here. So what is going on? Well, I'll just read it, right? So it's GCCT here, and then this is the duplicated sequence, and then it goes CTAT, and the CTAT's here. So this is where the transposon looked at. It cut this part right here and inserted itself. Note that this is just a 5' strand. If we had the 3' strand, the what you do is you make the cut here, and in the 3' you make the cut here. So this is GCTT. In the 3' strand, it would be CGGAA, and it would be over, and then it would make the duplication of it. If you're kind of having a hard time following, I have a picture for this. So there's also another thing I wanted to note, like, so we have this, the duplicated sequences, which is the most important part. So here, what I'm about to say, and when I'm about to talk about stuff that's probably not going to help directly with like a test, I'm going to say it, and this is like one of the times, this is more for like trivia and like pretty cool stuff. Another way to detect that this is actually a transposon that happened is to look at these. All right, so I saw this, or like I didn't see it eventually. I didn't even notice it because I had to look at the answer key or whatever, right? But this and this are very interesting, right? Because you put them over here. Well, they're not equal in length. Maybe if I just like go like that, whatever. Okay, so what I did is I got this point and I got it here, and then I got this and I got it here. All right. So then after I did that, I got this and I went online. I looked up reverse complement, you know, DNA giver, all right, like calculator or something. I plugged in that and it gave me the reverse complement. What's the reverse complement? What you do is, this is 5' to 3', right? 5', 3', whatever. The reverse complement is you flip it, right? You make this the 5', right? And then you get the complement. So you change all the letters. So this is 5. If I want that to be the 5', that's going to be a G, which is here. Oh, wait. Oh, wait. What am I saying? So you see what I did, right? I've made some sort of grave error. Oh, foolish me. All right. I shouldn't have deleted that in the first place. I'm silly. All right. So I see CCC and then GGG and then A, C, T, C, T, T, C, T, and so on. So what I identified here is that there is a reverse complement, you know, right downstream. What does that mean? Well, that's just like an indication that, you know, it's a certain type of transposon. And you don't really talk about like, you know, different types of transposons in 514, but it's just an interesting little like piece of thing. And I have this little quote here. There are two types of them, long terminal, or two types of class 1. There's different classes. And so for class 1s, there are these guys with long terminal repeats, and then non-LTRs, which lack the repeats. And so this would be, this is indicating long terminal repeats. So what I mean here is going down here. So this is like the mechanism of transposition of just a conservative, I think like cut and paste situation, right? So you have the transposon, and the inverted repeats are here. And those inverted repeats are these things. But again, this is stuff we don't need to know as much, like those inverted repeats. What you should be familiar is this. So this is the mechanism of duplication, like the two direct repeats right here. So this is just how it works. This is what I was trying to say earlier. Like you have a cut here and a cut here. You take them apart like it does here and here. You put the transposon in, and then it does just the normal like polymerization of filling in the DNA, and you just get 2 ATGCA. So that's an easy way to determine what the actual transposon is. It's everything in between those duplicated sequences and why that happens, right? Again, just another representation of why that's going on. Let's see, what do I have here? Oh, this is a cool little piece of trivia I have here. So L1 going back is like line 1. It's a type of transposon that doesn't have these inverted repeats, that doesn't have these inverted repeats. Remember that this is usually double-stranded, so this would be, whatever. It would have inverted repeats, right? Hemophilia can be caused by a transposon going into this gene. Pretty cool, right? Interesting. Whatever. I want to also, so that now we're going, this is like another kind of part where it's like a little trivia-ish, but also it some of this information can help. When you have, so I talked about like these inverted repeats here, which come from this piece of like this length of DNA here, and then its reverse complement being downstream. All right, cool, whatever. If you have a shorter version of this, or maybe it could be that long, but if this is like very close here, what you could actually end up getting is this part can fold in on itself and then make a hairpin. So hairpins are really are when you have like the reverse complement of some sequence of DNA really close, and it'll go like that. Hairpins are pretty cool, because they do stuff. All right, so what stuff do they do? Well, you may recall from your introductory biochemistry course with the fantastic Dr. Harris, something called road-dependent termination and intrinsic termination of transcription. So in bacteria. So in bacteria, there has to be a way, and eukaryotes too, but in bacteria I'm talking about, a way to end transcription. So a way to end transcription, there's two ways. There's intrinsic termination, which happens most of the time, and then road-dependent termination. I'm going to ask a question here. Between the two, which one requires a transacting factor? You may think of that for a sec. Give it a second. It's a pretty easy kind of question, but the idea is you want to really be able to start telling like being comfortable with the terminology transacting versus cisacting, because that's very important. So yeah, in the case of intrinsic termination, what happens is the nascent RNA is going to it's going to get synthesized by the polymerase. And at the end of the polymerization, it's going to make a sequence of DNA. And then really close to that, it's going to make its reverse complement. So it makes a hairpin. Now, there are a few models, these guys right here, on why that happens. You don't need, again, this is more trivia. You can look these up if you want. They're all made like the thought of in like 1980s and stuff like that. Like, it doesn't seem like there's like a set scheme onto how the hairpin itself breaks out of the ribosome. Maybe there are out of the polymerase. Maybe there is. I don't know. I don't know too much. I just like read this on like last night, so whatever. But right after the hairpin, it makes a poly-U tail. All right, which is pretty cool because what happens is that poly-U tail helps that polymerase break free. All right, so that polymerase can break free of this because of the poly-U tail. Because remember, uridine and U and A, they have 2 hydrogen bonds between each other. G and C has 3 hydrogen bonds between each other. So that means the bonds, the strength between U and A is weaker than that of GC. So if you're trying to take off a nascent RNA strand, all right, what you could do is or what the cell does, right? What the bacterial cells do is they make a lot of U's so that there's a weak association here, and that along with the hairpin helps rip off that piece of RNA from the RNA polymerase, and then you terminate transcription. Pretty cool. All right. So remember, this is intrinsic because basically nothing's going on here besides the fact that the RNA polymerase is just transcribing. The RNA polymerase doesn't know if it's like, about to terminate. It's the RNA, how this is folding and whatnot is going to make it go off. Rho-dependent termination requires Rho, which is a 6-membered helicase, which basically, upon seeing a little like little box here or whatever, like a little site on the DNA or something like that. I'm a little rusty with it, but what happens is Rho will detect that, or whatever, and it'll come over and then yank out the DNA strand physically from the RNA polymerase, which is also pretty cool. So between these two, which one requires a transacting factor would be Rho-dependent termination, because what happens is this termination needs the Rho helicase, which is a transacting factor. It's not on like the DNA molecule or whatever, If you looked at the DNA molecule, the cis-acting factors would be the things that the Rho directly interacts with, what like notes. So I think, yeah, there are both cis-acting and transacting factors in Rho-dependent termination, but none in intrinsic, because there's no like sequence of DNA that is like specific, whatever. It's intrinsic to the RNA molecule, that why it terminates. Okay, cool, whatever. So we're talking about that here. So right here, RUT, that's Rho utilization. All right, and that is the cis-acting sequence, right? These are the cis-acting factors for Rho-dependent termination. All right, so I have something pretty interesting. So one of the most studied Rho-dependent or Rho helices that that it is studied in biochemistry is actually the TR1 transcription termination like area, which is part of the lambda. So that's, I don't know, I thought that was pretty cool. Like so lambda uses a Rho helix, right, helicase to terminate transcription at a certain state. So let me go over. See, I say Rho plays a central role in the life cycle of E. coli bacteriophage. This is supposed to be lambda. As a key function in the regulation, the TR1 is one of the best studied Rho-dependent termination. Okay, whatever. Let me go take a step back, go to this little slide area. Okay, so here, this is like the lambda, like part of the lambda genome. And so we're kind of familiar with this stuff, hopefully. So if we look here, TR1, which is in between CRO and C2, this is where the helix will go and stop termination, right? So it would be of the PR. So in the PR, starting to transcribe here, we look here in this area, that's where it would stop transcription at the nut R and then the TR1 here. So if there's a way to bypass that, then you could induce the lytic cycle, right? Because usually when you go past this, all right, these are like the PEQ, SR, like that stuff makes like the capsid head and tail and stuff like that. So that's lytic, onset. So what N does is it binds to this area and then something like that, and it yanks out like that's not the helicase, but it has something to do with it. I don't know. Whatever here. This is a really cool thing I found here. Don't want to talk about this now. This is just like, again, this is another visualization of the genome of lambda. But also it has these like colored things, which are really cool. So at the blue stage, it's like 0 seconds, and then everything up is like 2 minutes more, right? So as soon as lambda infects a bacteria, what happens is these things are, if we look around the whole thing, looking at blue, are the earliest like things that are being expressed, and we know that for like C1 and CRO, right? And these guys are like kind of competing, right? Whatever wins determines lytic, lysogenic, whatever. But we also see Rex A and Rex B, and we've never seen these before. These are pretty interesting. This is more, again, like trivia-ish. Rex A and Rex B are really cool, I thought. And they're just like, we go here. So Rex A and Rex B, what they do, they're very, so again, trivia, so don't commit it, like don't go crazy with this, right? But they're like genes that also promote lysogeny, right? And so if you go up here, this is Rex B, this is Rex A, right? What they do is they interact with each other, and then it causes this depolarization right here of the membrane of the bacteria. And this depolarization will sometimes, it'll like kill the cell and stuff like that. So what's the purpose of that? Like if a bacteriophage like is about to endure the lytic cycle and it doesn't want to do that, I guess like it expresses these and then it kills the cell instead of becoming lytic or something like that. Or maybe it's more so that like it's more so that maybe this is a lysogen itself. I'd imagine this is a lysogen itself because you have these proteins already, translated in the membrane and whatnot. So there's probably a prophage, a lambda prophage in this already expressing Rex A and Rex B. And so this is another, guy coming in trying to infect it, but this is like, nah, go away. And then it's like, I'm not even dealing with this. I'll die. So it'll kill the cell, maybe. I don't know. I just thought it was cool. And it's one of the only, few guys that are being translated first. So all of this stuff, like you could literally just look up and get interested in. Let's see, TR1. So yeah, this is TR1 is like what I was talking about earlier right here. This is like the area. If we look in, it's kind of hard to see, but this is Crow. All right. And then this is, wait, no, that's Crow. Oh no, this is the end of Crow. So Crow is the end here. And then this is C2 right here. Crow and C2. So in between Crow and C2, you have this area, right? Which, you have with which has to do with the rho helicase stuff. So the rho helicase will go to rut A and rut B basically. And those are the cis acting sequences that help the helicase note that that's where to terminate, like transcription should terminate. Box B is where the N protein itself binds to the DNA, like this. These are cryo-AM. So this is N right here. It's a short polypeptide that binds to the DNA here. And once it does that, I have this little thing here. It binds as predominantly with major groove. Whatever. It doesn't say what it does. I think what it does is it allows the allows the read through of this right here. So maybe N by binding there, it makes it so that the rho helicase can't bind to rut A and rut B or something like that. So what the reason why I kind of brought up rut A and rut B is because I found this cool little paper. Oh wait, no. Oh no. Got confused. The reason why I talked about Rex A and Rex B is because I found this cool paper talking about like a T4 bacteriophage. So you have lambda bacteriophages, and T4 bacteriophages are a different type. And they have a different genome. And the thing with those is it's impossible for them to do lysogeny, or we think it's, normally it's impossible, right? They're a purely lytic bacteriophage. But these guys, what they did is they subcloned Rex A and Rex B, put them under the control of these promoters. Cool, interesting. And they demonstrated that they're sufficient to restrict the growth of T4 R2 mutants, which is, that's, if you want to look that up, that's an interesting concept in itself. Yeah, it's an interesting concept. There's crazy stuff going on with that, whatever. So yeah, basically what we're seeing is still like this, like T4, like basically immunity against T4. All right, which is usually not what happens. T4 usually goes in and just causes a lytic situation, and without C1, right? So whatever. This is just all information I'm throwing at y'all. A little thing. Like, yeah, the N protein eliminates termination events, permits transcription range of early genes. All right. Sorry, I had to look something up. All right. So we go to, there's another question I want to talk about. All right. So we just went on this little rant, whatever. you know, yada, yada, yada. I don't know if y'all are into this or not, but I am. I'm having a fun time, whatever. So this is another question from that test, that MIT test, which is completely different. Some of this stuff, like you won't get a question like this on your test, but like Dr. Myers likes to ask like throw diagrams like this at you, and it's good to be able to like not be too intimidated by like looking at that, like what this is, or maybe even like this. All right, so I feel like this would be like a good kind of question to walk through. All right. Yeah. So let's just read it. All right. You're studying an expression of E. coli ribosomal protein that inhibits protein synthesis by bonding to short RNA hairpin. Cool. We talked about that. And there are ribosomal RNAs. This protein, very low nutrients, whatever. It's first step you perform a northern blot. All right. So you might not know what a northern blot is, and that's completely fine. You know, Western blot is when we look at proteins. A southern blot is when we look at I think, right, like DNA, right? And then a northern blot would be looking at RNA. And the way they do this is you look at the RNA transcripts by making a DNA like a piece of DNA that'll hybridize with that RNA transcript, and the DNA has like, you know, you're able to look at like you make the DNA, like you augment it with like something that makes you able to see it on a gel. All right, so that's what a northern blot is. You're just looking at RNA expression. So that's all you need to know. Determine the rest of the, I don't know if this is whatever, you know, these are just words. I like to look at the diagrams and stuff like that. So basically what's happening is we have 3 genes. All right, we have this guy, this guy, this guy. And so we have this going on. So what this is 3 northern blots, each with a different DNA probe. So this DNA probe is the DNA probe, like if there's a transcript for this guy, the DNA probe for this will attach onto that, and then see how big that like signal is. These are nutrient plus minus conditions, so don't worry about that too much. I'll talk about that at the end, and it'll make sense. So just the question is for 4A, based on the results of the northern blots, how many different mRNAs are synthesized that overlap DG1, RP28, and CH1 genes? Okay. I'll pause the video, or pause the video for a sec. Try to figure that out for yourself. It's pretty easy. It's just how many different types of signals are there. All right, welcome back. You know, maybe you didn't go, whatever. So there are 3. All right, so we look at the plus minus here. It doesn't seem the nutrients don't have any sort of deal on the GG1 expression, right? So they're the same. It's around 600 base pairs, right? So like that makes sense. Like this area, that's what's being transcribed here. If I go here, right, I have 2 signals, right? 2 different signals, and then 1 signal between these 2. These guys share the same signal, so there's 3 in total. 3 transcripts: 1, 2, 3. 1 is 600, 1 is 1000, and this 1000 one has a DNA probe of CHW1. So this is going from like here to 1000 here. It has some of these like flanking regions. Okay, cool, whatever. And then we have a long one, 2000, that is picked up by this DNA probe and that DNA probe. So it's probably going from like 0 to 2000, right? That transcript, and the DNA probe for this and the DNA probe for this can both hybridize onto it, and then, you know, kind of like, you know, show it, whatever. So yeah, there's 3. Okay, interesting. So we basically just talked about where those were, you know. Yeah, cool, whatever. So here's this interesting question right here that I talked about, row dependent helices, and those 2. I reminded you all of that for this question because I thought it was cool. You find that there's an intrinsic terminator at the very near the 3' end of the 2000 base transcript. So here, from here to here, over this area, the transcript for that is going to have an intrinsic terminator, which is that hairpin, right? In contrast, you don't find that near the end of the 1000 base pair, which is pretty interesting. Around this area, you do not. What you see is a C-rich region. Cool. What does that mean? So, you know, C-rich region is another indication of a row dependent termination, right? So what's going on here is for the 1000, right? For the 1000 base pair transcript for this guy, at the end of it, it got terminated by a row dependent helicase. This 2001 that ended here got taken over by, or got it, stopped by an intrinsic terminator. The thing is, though, think of it. The 2001 like the transcript for the 2000 has the bypass, like once it's transcribing from here to here, it has to bypass that row dependent helicase termination step in order to get here in the first place. So that you should be thinking, why does it do that? And now here we got some postulation going with, you know, with respect to the nutrient rich and nutrient poor conditions. So it's saying, yeah, using your understanding of the row termination factor and whatever, propose a model that explains the presence of the larger transcript in the presence of low nutrients. So this is pretty easy. You look at how row dependent termination happens and how intrinsic termination happens. And so if we look here, let's look at RP28. I mean, no. Yeah, RP28. So it seems like at RP28, there's no, like, I don't know, maybe there's like no promoter region here or something like that. Like the expression of this seems to be controlled basically by the promoter here. All right. So in order to get RP29, you know, in order to get RP29, what you do, oh my goodness. To get RP 28, you have to start the transcription here. It has to read through under nutrient poor conditions. CHW1 in nutrient and good nutrient conditions, you know, has the row dependent termination, but when under lower conditions, it'll have this, the 0 to 2000, which contains both the CHW1 and the RP28 segments. So the hypothesis, so the idea is, you know, the overall idea, I guess, the answer is basically that row dependent termination definitely requires more energy to do than that of intrinsic, because intrinsic, it's literally like the DNA, the RNA segments, you know, making the hairpin and then the poly U tail, like that requires no energy other than like polymerization, right? Because that's just like intrinsic to the RNA piece. Whereas row dependent termination requires a transacting factor, which you have to make. You have to synthesize, all right, or not you, but like the cell, right? But it synthesizes that. And it also requires energy to pull that, as a helicase, to pull out a piece of RNA from a RNA polymerase. So there you go. Another kind of situation going on that requires energy. So yeah, that might kind of make sense. You also note here, it says you're studying expression of E. coli ribosomal protein, RP28, that inhibits protein synthesis by bonding to short RNA hairpins. So when we have low nutrient conditions, it seems like RP28 is expressed. All right, cool. So what does that do? So as soon as RP28 gets expressed, it seems to bind to hairpins. All right, hairpins are found over at, we said the hairpins were where? Here at the end of the 2000. All right. So that's also interesting. So the RP28, upon being made, it'll bind over there, and I don't know exactly. You know, I forgot. So let's just look at the answer, which I put here because I expected I would not get it completely like right. So here's the answer for it. It's pretty interesting. I would read it for yourself. Try to digest it on your own. But it says row must be down regulated in low nutrient conditions because there is less ATP available, right? And there's like some sort of positive feedback cycle going with RP28, like inhibition or something like that. It's like inhibiting itself or something like that. I don't know. Read it. It's pretty cool. Interesting. But yeah. Like pretty interesting stuff going on here. Those are like the two questions I kind of like found that I thought would be interesting. And then like, you know, going over this stuff was like cool. So I don't know. If this, you know, interested you, let me know. If you're a fan of this, let me know. If you want anything, you know, talked about in particular, let me know. Yep, I'm here to help y'all out. And yeah, this is pretty fun. I'll catch you later. How do we stop the recording? There we go.